So I don't really do quantitative genetics much, and I definitely don't do philo thingies, genetics. Whatever, you know. But I have done a little bit. So I'm just going to talk about a couple of the things over the years. What I usually tend to focus on is how the fact that species are separated from each other, individuals in a species are separated from each other by physical space, and particularly when that is accompanied by some areas that are good habitat where the species can be at high frequency, and other areas where they might be at low abundance or absent. That kind of population structure generated by space, what are the patterns associated with that, and how do those patterns affect the way evolution can happen in those kind of populations? So that's kind of my framework on this. I want to talk a little bit today about how that affects quantitative genetics. And so here's kind of the roadmap of where we're going. I want to talk a little bit about how we can understand the partitioning of genetic variance within and among populations. And I want to do that in particular with a focus on neutral traits. And the reason that I want to talk about neutral traits is not that that's what I think is the most interesting kind of traits. Most of us got into evolutionary biology because traits actually do stuff. But it's necessary to have a good null model of the way evolution works without that selection in order to infer. And the kind of scary thing is how strongly patterned neutral variation can be. So these null models are a lot more potent than would be nice. It'd be much nicer if everything was exactly bland and gray without selection, but that's not true. So I want to talk about one of the measures of looking for local adaptation, responding to selection differently across a heterogeneous spatial landscape. And then at the end, I'll switch gears and talk for like 4 or 5 slides about the only paper that I wrote that Steve can remember about migration and how it affects the Gmatrix. It's actually it's good. I've only written 2, so that's okay. So one of the things that we're going to need today comes from sort of basic population genetics, and so I just want to spend a couple minutes talking about it. And that's this thing, FST. FST is a standardized measure of the genetic differentiation among populations. It was invented by Sewell Wright 65 years ago, and it's in common use. One of the nice things about FST is it's a thing we can measure from the kind of data that we can get from genotyping individuals. And there's a lot of different conceptual interpretations of this quantity FST that turn out to interrelate in some neat ways. So the kind of classical way of thinking about what FST is, it's a measure of the variance among populations, a standardized measure. So if we looked at the allele frequency, this is just at a single locus level here, no quantitative genetics. If we look at the allele frequency of a single locus and measured what that was for multiple populations, I took the variance of that list and then sort of standardized it by the average allele frequency times 1 minus that average. That's an interpretation of FST. It tells us something about how much variance there is among populations of allele frequencies. Another thing FST is, broadly speaking, it's a measure of the probability of identity by descent of alleles that are in the same population. So if you take 2 alleles and you ask, are these the same over and above what you would expect just by looking at the overall species allele frequency, FST tells you that probability of them being identical by descent. Okay? So from that, it tells us about excess probability of homozygosity of an individual in a population. That's not an aspect we'll talk about much today, but it is a measure of the common ancestry of the alleles as well, and that'll be important for us. And there's another interpretation of FST closely related to that, which I'll come back to in a second. So when I say FST, it's a standard population genetic measure. Everybody, have everybody heard of FST before? Steve asked, good. Okay. All right, so here's just a few little background factoids that we'll put together here in a second to talk about putting quantitative genetics in a spatial context. One quantity that turns out to be really useful is to think about something like a pooled variance. I don't even have a good word for this, but how much quantitative adaptive variance would there be if you took all the individuals in a population and all their alleles and just mixed them together into a population? It removed the population structure entirely without changing the overall allele frequency. How much additive genetic variance would there be? And that's just a simple function of the allele frequencies, and it doesn't have to be a 2-allele case, but it's easier to write. That's just a function of the allele frequencies in each population. These are I for each over each locus, the allele frequencies and their average effects. Now, I'm going to give you a big caveat. Most of what I'm going to be talking about for today is a very simple case, and that's the case where alleles interact additively, right? So that we can just take an allele, we can put it in any individual, and the change in the phenotype that allele would cause by adding it to that individual will be the same regardless of the rest of the genotype of that individual. So there's no dominance, there's no epistasis. Okay? That is actually a crucial assumption that will change some of the things that we'll talk about later, but that's the starting point, a good starting point for thinking about these things. So that's the assumption we'll be making for at least the first 3/4 of the talk. Okay, so we can just mix these things together. We can measure how much genetic variance would there have been in the absence of population structure. Okay, now Sewell Wright, and I can't remember the reference for this, but it was in the mid-40s, showed that we could actually predict under this circumstance that I was just talking about with when alleles interact additively, we could predict the amount of added genetic variance within a population by knowing what that overall pattern of genetic variance would be in a panmictic population and multiplying it times 1 minus FST. Okay. Now this kind of comes about intuitively. You can imagine this comes about because there's, remember FST is a measure of the identity by descent of the two alleles that are in the same population. And so to an extent FST, two alleles in different individuals in the same population are actually the same. They're not different from each other in their effects that they're causing on the trait in proportion to FST. Okay? So the variance among individuals is decreased within populations because of the shared descent of the alleles that those individuals carry. For diploid populations, and this should say diploid somewhere on the slide, for diploid populations, the amount of genetic variance among populations, right, calculate the mean of each population for its traits, measured in some common standard environment, and then take the variance among those means to calculate this genetic variance among populations. That turns out also, of course, to be proportional to the amount of genetic variability in the population, but times 2 FST. Okay. That 2 actually comes because they're diploid. If they're haploid, the 1 goes away. It's actually coming here because another interpretation of FST is it's the covariance of alleles in the same population. And so when you have population structure, the two alleles that are in the same individual are more likely to be the same as each other and they covary in respect to their differences from all other populations. And so that covariance generates actually extra variance among individuals in total, right? If you add these together, it turns the 1 plus FST times this VA naught. Okay? So there's more genetic variance among individuals in a diploid structured population than you would have found in a panmictic population. Okay? And the extra bits are showing up because of this covariance among populations. So Wright showed this to us. We can predict the structure of genetic variance. I should say, I've talked about the assumptions that we're making in terms of additivity. There's another assumption that we have to bake into this because it's what we're trying to generate. We want to generate a null model for what happens without selection. And so here is the adaptive landscape that I'm using, right? It is perfectly flat. right? There's no lines on the contour plot here. So we're just dealing with neutrality, and that's because, as I said, we want the null model. So we'll relax that again later. Okay. Mike Lynch and Bruce Walsh, several years, no, they didn't show this. Well, Lynch and Walsh talked about this a couple of decades ago, that one way to predict under these assumptions of additively acting neutral genes, how much genetic variance would there be in a population, could be predictable by the effective population size of that population and the amount of mutational variance input to that population per generation. Okay? So this is just, again, banning selection, a mutation drift balance for a single population. And what I want to do now is kind of expand this a bit to think about how this acts from related concepts in a structured population. Now, one of the key things that you might think here is this 2NE is actually, there's two ways to think about this. On the one hand, this is the inverse of the amount of drift that's happening. So the higher the NE is, the less genetic variance we're losing per generation due to fixation. Another way of thinking about it, though, is that this 2NE for a diploid model is the average time that two alleles, the average time that back in history for the last common ancestor of any two randomly chosen alleles. Okay? So in other words, on average, the shared evolutionary history of two individuals in a population were the same 2NE generations ago. And so there's been 2NE generations of, on average, of accumulating differences between individuals as a result of that phylogenetic history. Okay? And that's the kind of property that we'll explore. Any questions? Okay. Well, if that was a I'm too confused to ask a question or you're going too slow, pick it up, I don't know. All right, so here's a phylogenetic history of a population, a single population, the coalescent history. This is a graph probably you've seen something like before. Here is the present time. Here are particular alleles at the current point in time. And those alleles will share, any two randomly chosen alleles will share a common ancestor at some point in the past. These two shared a common ancestor relatively recently. This one and this one, it's way back in time. On average, as I said a minute ago, on average, the time to a most recent common ancestor of a pair of alleles in a diploid population with effective population size 2NE, effective population size of NE, is 2NE generations. Okay. Now that's for a single locus. You can take that same phylogenetic tree and you ask not just what's the actual history of ancestry, but how different are the individuals on this tree. They become different from each other to the extent that their mutations happen in those particular alleles. Okay. And so these two, even though they share a common ancestor not immediately ago, because no mutations have happened, they'd be genetically identical. This one and this one would differ by two mutations, et cetera. So on average, the amount of mutations that accumulate is proportional to the amount of time that separates two individuals. And so it becomes, it generates the process by which this genetic variance occurs. Now this next slide is kind of a cartoon. With quantitative traits, if you imagine thinking about the particular effects of a single locus on a quantitative trait, that kind of variation could also accumulate in a similar process. If the more time you've had, the more mutations have occurred to make those alleles different in their effects on a particular trait. Now this is not what it would look like necessarily for the trait as a whole. The tree would be different for each different locus. But this is what the effects of a particular locus on the phylogeny, on a particular locus would be on a particular quantitative trait. Now, from the point of view of figuring out on average how much genetic variance there is in a population, we don't need to know the details of this phylogenetic tree. we just need to know the average amount of time that separates pairs of individuals in a population. So by the same thought process that I talked about a minute ago, the genetic variance in a population is going to accumulate in proportion to the amount of genetic variance between individuals that occurs for each generation of mutation times the average number of generations that separate those individuals. Okay. Now, this does not have most of the cool properties of coalescent trees, where we can understand the full distribution of what we might expect to see about the differences between individuals. But it's a good predictor of the amount of genetic variance on average in the population for a particular trait. Okay. Well, let's put this in a population genetic context, into a multiple population context. One of the other interpretations of FST came from Monty Slatkin in 1991. Turns out to be very useful. So another interpretation of FST is that it is giving us an index of how much shared history there is for individuals that come from within a population compared to individuals that are drawn at random from the host species. Okay, so this T species is, on average, how different are two alleles, how on average, what's the time in the past to the most recent common ancestor of two alleles when those alleles are chosen at random from the whole species. And this T within is the same thing. It's the average time to the most recent common ancestor when the two alleles that you're looking at came from the same subpopulation. Okay. Now, alleles that come from the same subpopulation are on average going to have a shorter time to a most recent common ancestor than individuals that are in different populations. And a good way to intuit that is to remember that in the kind of population structure we're talking about, it's very unlikely for individuals to be able to mate with an individual that's in a different population. Okay? So in order for alleles to have in order for two alleles in different individuals to have a common ancestor, the genealogy of those alleles have to date back to the same individual, and that same individual has to be in one place at that time, right? So individuals that are in the same, two alleles that are in the same population have already, thinking anthropomorphically, they've already solved one of the problems about having a coalescent, right? They're in the same place. And so the alleles could have been drawn from the same individual because they're already there. Individuals that are in different locations, not in the same population, going back in time, there's an event that has to happen first before there can be a coalescent event, and that event is the two ancestors have to actually be in the same population. And only then are you back to the point where the amount of time it takes to get coalescence is the same as individuals that were chosen from the same population. Okay? So basically the difference between this and this is how long does it take, going back in time on average, between where you are now and how long it takes for a random pair of individuals to get into the same population. Okay, so this is always going to be a positive number between 0 and 1, just like F, well, it is FST. Okay, so there's a coalescent interpretation of FST, and that turns out to be useful when we think about quantitative traits, for reasons I'll show you in a second. So the next block of slides, what I'm going to try to do is introduce a measure, QST. How many people heard of QST? 50-50? Not? Okay. So I'm going to introduce this QST. The main goal of this is to show you that QST for a neutral trait is on average going to be the same as FST under a certain set of conditions. And then we'll move to the next part of the talk and talk about the strengths and weaknesses of actually using that QST in inference. Okay, so here's the QST. Here's the definition of QST. It's a property of the variance among populations. Here it says variance within. It would be better here if this said VA. So this is the additive genetic variance within populations. Okay. It's amazing what you see when it's on a big screen that you don't see on the small screen. So this is a measure of genetic variance within populations, but specifically the additive genetic variance within populations. This was originally defined by Ken Spitza in 1993, and also by Tim Davis, somebody else, huh? Prout, yes, thank you. Tim Prout. 2 papers that came out in the same year. Now, this kind of looks like a sort of arbitrary definition here. If you look at it a little more deeply, it seems a little weirder. Like on the bottom here, it's variance among plus 2 times the variance within. Why is that? It turns out this is the equation that comes from diploids. Okay? With haploids, that 2 disappears. So for diploids, there's a 2 there, and that appears for reasons that hopefully I'll try to make clear as we go on, but maybe not so explicitly. It comes from the fact that there's actually the variance among populations is proportional to FST, whereas the effect on variance within populations is proportional to 1 minus FST. So that 2 in the variance among winds up in the algebra landing in there. But I'll try to show you how that comes a little bit here. Another way to rewrite that is this. Okay? If you took each of those quantities, V among, V within, and used the same equations from right that I showed you on like the 3rd or 4th slide, you can quickly get to this kind of formulation. Okay? Please. No, V among is it's not there's not a breakdown of additive and dominance variance that I'm aware of for among population variance. What it is if you when you take those genotypes and you grow them in a common garden, it is the variance among those populations in the means of those populations. So if we had not already made the limiting assumption that we're talking about only additively interacting alleles, that would also include dominance differences, et cetera. Okay. Right. So these variants at the species level, the variance in the within population level, as I was just telling you before, those turn out to be functions of the average amount of time to common ancestors of the alleles, either in the species, 2 randomly chosen alleles from the species, or 2 randomly chosen alleles within population, just like this. So within populations, there's this much time, right? This is the average time to a common ancestor within populations, and that's how much time you've got to accumulate differences between individuals. At the species level, same kind of thing. Okay, so we can put these back in to the QST, and you recreate this this equation here, which should look familiar. It's a function of the average time of the species level, the within level of the coalescence times. That's the same equation that we have from Slatkin for FST. Okay, so we made a lot of assumptions here about the nature of interaction of these alleles, i.e. that there wasn't any. We made assumptions about selection, that there wasn't any, but we did not make any assumptions about any of the demographic details of how these populations came to be different from each other. Okay? So what this means is that we under these conditions, for neutral additive characters, QST should, on average, equal FST. If you're actually making notes on something, you might want to add in actually E bracket, close bracket around QST and around FST. This is a statement about what FST should be on average versus what QST should be on average. Under these conditions, they should be equal. That distinction we'll make much of in a second here. Okay. Right. Now, so we're going to segue a little bit to why we might care about something like QST. The reason that we might care about QST is if you are concerned about selection, in particular the kind of selection that causes some traits to be favored, or some values of traits to be favored in some localities and other values of the same trait to be favored in other localities, In other words, spatially heterogeneous selection, or you might call it local adaptation. Under that local adaptation process, there is an extra reason that the populations should be genetically different besides just drift and mutation. And that reason is selection. Selection is causing those populations to diverge. And so, if you have spatially divergent selection, spatially heterogeneous selection, you might expect to see that QST is not equal to FST, but actually greater. Okay. So that's the kind of promise here. So FST is something that's very straightforward to measure. QST is, in principle, straightforward to measure. In practice, it's a pain in the tuchus, but we'll talk about that in a minute. But we do have access to the information that would allow us to make this kind of comparison. This would tell us nothing directly about the nature of that selection. It would just say this trait, if we successfully showed this to our satisfaction, it would just tell us that this trait is under some form of spatially heterogeneous selection, and there would be a hint that we might go do other biology to pick that out. Yeah.
Maybe you're going to make this point, but if we were predicting that there would be spatial uniformity in the same pattern of stabilizing selection, would we predict this expression with the inequality reversed?
Yeah. So if you have stabilizing selection for the same optimum everywhere, this would be QSTs less than FST. The reason I don't highlight that is there's some biases in QST that come when we add non-additive genetics later that go in that direction and make it look like that anyway. So there's that plus some statistical sampling pains make that direction actually a less useful, a much more problematic comparison. But yes, true. In terms of understanding what this means, absolutely. Yeah. Okay, so we've been assuming non-additive genetics all along, and here's just a little glimpse into, this stuff gets just grievously more difficult when we get rid of that assumption. But that's been analyzed a little bit. If you have epistatic variance in the form of additive by additive epistasis, that will reduce QST relative to FST. Okay? Not necessarily by a lot, but in that direction. Okay? And this is on average. Nobody's looked at other kinds of epistasis besides additive by additive epistasis. So what would additive by dominance epistasis or dominance by dominance? The answer is I don't know. My guess is it would be complicated. In some conditions it would go up and some down. But what the preponderance of the effect is, we don't know. So if you're looking for something to do that would take you the next 3 years of your life, go for it. Dominance variance, though, we do know something about. So dominance variance, and when I say dominant, I'm mixing two things here, but dominance variance or when there's dominance in the form of interaction between the alleles, regardless of whether it creates much dominance variance or not. Dominance variance can either increase or decrease QST. Usually, and by usually I mean on average, it decreases QST, but it's by no means across the board. It depends on the circumstances. So essentially, if you have situations where the recessive allele is likely to be more common on average across all the loci that affect the trait, QST will be lower. If the dominant allele turns out is usually the common one, I'm getting this backwards, sorry. If the recessive allele is rare, it tends to decrease FST. If the dominant allele is rare, it tends to increase QST. Did I say all those words in the right order? I don't think I did, but right? So there's a paper by Jérôme Goudet and somebody, Buckley, I can't remember his co-author's first name, in genetics about 4 or 5 years ago that goes through extensive calculations about the full range of combinations of different loci. So the short version of this is you won't know for sure what dominance variance is going to do for your trait, probably because the whole point of doing quantitative genetics is we don't know the kind of details at a locus level that you actually need to know to predict this. Okay. If you have a trait where If your trait was fitness, then you can make predictions that the recessive allele is likely to be the rare one because of the nature of the way dominance usually works in terms of fitness. But that's not easy to do for quantitative traits in general. Okay, so everything I've talked about at this point is about additive only traits. The real world didn't read that book. That's not the way things work. Epistasis, probably not a big problem if we're looking for local adaptation. Dominance variance is more difficult. It's probably not that big a deal, but in any particular case, we don't know for sure. All right. Okay, so just stuff we just said. FST can help you predict the structuring of quantitative genetic variance. We've known that since right. And this one's getting a little bit ahead of ourselves, but if you looked at the comparison between a measure of QST and a measure of FST, we can try to infer something about the strength of spatially divergent selection on the trait. Okay, so let's talk about actually the challenges of using this QST if you ever decided to do so. One of the biggest challenges is that both FST and QST have the potential for extremely broad sampling distributions. Okay. In the case of FST, that's not such a. Well, let me just tell you what these graphs are. These are from simulation, from in this case an island model where every population is equally connected to all the others. Everybody's the same size, et cetera. So this is our kind of starting go-to model for population structure. This is a distribution of what individual neutral FSTs values would be for different loci. This is a distribution for a polygenic trait QST value. So each of the elements here is the QST of a given trait, made up in each case of lots of different loci. Now, the thing that most of us find counterintuitive is that the breadth of the distribution for the QST of traits is just as great as the breadth of distribution for FST for individual loci. And I can't easily explain that to you. I have some glimmer of it, but I'm not going to try to put it into words. It's the kind of thing, I've simulated this a lot over a lot of different circumstances. I'll show you a bit more in a bit. And it is true, the distribution is similar, or almost the same, for individual loci versus individual traits. Okay. So the problem is, we don't care in this context about all those individual loci. We can measure now with genomics, you can measure 40 squigillion individual loci and get a really reliable index of what the average FST is. Right? So that's what you need to make the comparison, is what's the average FST. That part's not a problem. The problem is we are interested in individual characters. Even if you're looking at 10 of them, you're not wanting to average those 10 to get a QST. You want to know what's the QST for each of those traits, so you can ask, is this trait having spatially divergent selection? So we can't increase our power in that way for QST. There's this huge distribution. So this is one where the mean is 0.05, and you notice it goes effectively from 0 to over 0.1. I don't think I have the slide here, but for I've calculated graphs for if you did intercontinental FST measures or QST measures for humans. So there's effectively only 3 continents, and the number of different populations that you have is a big factor in how broad this distribution is. If you're limited because of the biology of the system to a limited number of populations, the range can be enormous. Right? So for a human population, the FST among continents is on the order of 0.15, and then the range of what you would see for a graph like this, even for the for the 95th quantile in the middle, would range from about 0.01 to about 0.6. Okay? So there's an enormous problem that comes from how broad these distributions are. Okay? And that's to get back to my point I kind of alluded to earlier, that even if you don't care about neutral stuff isn't listening to you. They're going to make you care. Because there's a lot of variation that you can see in nature, a lot of patterns that you can see in nature that are generated by entirely neutral processes that look a lot like, especially if you look at it naively, look a lot like what selection would do. Okay, so in this case, you could easily get a doubling of the amount of variance among populations, and it would not be statistically significant. In the case of the human among continent comparisons, you have to get greater than fourfold variance among populations before it reaches the zone where it would be statistically significant. So it's huge effects that drift could generate false patterns for. Okay. Why is this? Well, let me show you for the F. Why is it so variable? So here, just imagine that you have two different populations that are drifting around over time, and I've, in this case, chosen some fairly small population sizes just to show the effect. But depending on where or when you sampled this, you might have sampled them at a point where they, just by chance, one of the populations had drifted to a high frequency and the other would drifted to a low frequency, and that would have a high FST. Or you might have by chance sampled them at a point where they were similar, and in that case it would have a low FST. Now, we're not actually measuring things at different points in time, but when we measure different loci, each one we're catching at some point during some temporal cycle like this, and that generates some distribution. Now, you can imagine that if we had three populations, that we'd have a more consistent, slightly more consistent FST at any point we measured it. If we had 10, it'd be a lot more consistent. If we had 100, it'd be very consistent, right? So it's a big part of the statistical challenge here is that we often have only a small number of populations that either exist or that we care about or that we have data about, and that sets, that's going to set a lower bar for how much statistical power we can have. Okay, and a lot of times people apply these QST approaches to like subspecies comparisons, where it's just two things that come into contact, and they're looking at the differences between them, and that's the worst case scenario, right? So if you really only got a limited number of populations, you're going to have trouble with statistical power unless something's got a very, very strong effect of selection. Okay, I told you that I'd done similar simulations with other kinds of population structures. This is not all of them, but so over here is kind of cartoons for what I did. Here's a stepping stone model where things only exchange migrants with adjacent populations. And so here's the distribution of FST. Here's the distribution of QST for that scenario. And you can see they're very, very similar. What I've superimposed here with these graphs with the black lines, are not some kind of interpolation of a description of that actual distribution, but they are the prediction of the distribution of FST that you would get from Lewontin-Krakauer, assuming that the distribution follows a chi-square distribution. Okay, so you can see for the island model, you saw before with the island model, the black line describes it very well. It does remarkably well for that, even with these alternative population structures, but not perfectly. So here we have a kind of source sink model where there's a kind of core of large, long-lasting populations, but each one has some kind of satellite population that is small but gets a lot of gene flow from the core. So these kinds of populations are quite different from each other, and yet the overall pattern doesn't show much effect of that. You would definitely see differences of FST if you look just at the satellite populations. But in the absence of knowing that, you still my main point here is that the distributions of QST and FST are similar regardless of that. And here's an extinction colonization model. Right. Okay. So that's it. So not only are the averages under these conditions we're talking about, not only the average of FST and QST on average the same, but the distributions of FST and QST are very, very similar as well, which turns out to be a useful fact that's used in some of the analysis approaches that I won't really talk about much later. Okay. Questions? Right. So I want to segue to talk about a little bit about the problems of QST. One of the problems is it's actually hard to measure these things. And I say hard, I mean that in terms of just the physical labor point of view. You have to, when we talk about the variance among populations, you actually have to grow these things in a common garden. So you know that the differences between populations are not just phenotypic plasticity. If you really want to do it properly, you want to do that for a couple of generations so you don't have maternal effects that are plastic that are echoed in the generation that you measure. Okay? So it's a lot of work. You also have to do an experiment to measure the added genetic variance of the traits that you're talking about, and that's a fair bit of work as well. They can be done simultaneously, but it's a hefty project. You have to really want to do it. And there are certain species where you really, with any reasonable constraints about human lifespan and grants and stuff, you just can't get that information. Like if you cared about forest trees, it's really going to be difficult for you to get all the bits that you need to calculate a QST properly. Okay. So what some people have done is sometimes they don't. Sometimes they still call it QST, but just for clarity, it's better to call this thing by a different name. Some people have said, well, let's just measure the variance among populations that we see in nature. Right? There's lots of individuals out there. We'll go measure what they look like. We'll bring that data in. And we'll use the phenotypic variance instead of the added genetic variance. Okay, so that's way, way easier thing to do. And it's frankly a much more common thing for people to do. So this is something that you might call PST. Just there's no quantitative genetics in there, so get rid of the Q and put in P. The problem is, as I guess I've alluded to building up to this variance among populations is going to be an overestimate because there's going to be, well, typically an overestimate. There's going to be a lot of sources of variation that come from plasticity that make populations different, even if they're genetically identical. Okay? So that's going to make QST look bigger than it really is. Okay. And then if you use VP, that's an overestimate of VA, right? Because it's unless heritability is 1, VP is greater than VA. And so that's inflated by the environmental variance, and that's going to cause it to be an underestimate of what QST truly is. So if you look through the literature, you'll find papers that make both of these approximations, or that make, they do this in a common garden, but they don't have the VA. So their QSTs can be off, what they call QSTs can be off substantially. And so this, I just want to say that's a really bad idea. It's not worth probably your time doing it. And if you ever review one of those things, they don't do it. Right, so this PST isn't QST, and therefore it's not FST. And now the thing is, I think a lot of people look at QST and they say, oh, here's this arbitrary number. It looks kind of weird definition. Why can't we use just whatever measure we want? Why would we have to stick to that specific definition? Why don't we just do something that talks about how different populations are from each other? And I hope I've convinced you so far is that the reason is we have an explicit set of calculations that, at least under these assumptions, say that this should match FST. Not just that here's a thing that's kind of similar to FST in the way it looks like, but that it should, under these assumptions, actually match FST. And so if we calculate something that's not that, then we don't have any a priori reason to compare it to any particular thing. Right? So the derivation, I guess, actually matters. Okay, so here's a few more challenges, some of which I mentioned. The data is really difficult to collect. Non-added genetic variance is potentially a big problem. Dominance interactions between loci, you have to think very carefully about what kind of dominance interactions you might have, compare it to the Goudet and Buckley paper, and hope for the best, I think. A statistical challenge is that the expected error distribution of QST, error is not the right word, but it's just the distribution of what we might see under the null model, under the neutral model for QST, is extremely broad under most circumstances. And so we actually have to have to be very careful to try to include as many populations as we can. And we have to not go into this unless we're looking for fairly large effects. I guess that's the main advice. If you don't think that the QST, if you're not looking for big, strong differences between populations, it's not worth the effort. QST, this is an advantage and a disadvantage. We never said what the environmental axis was that separated these populations. We're not looking for what's causing, at this stage, we're not looking for what's causing this selection. We're just saying, is there evidence that some kind of selection has been effective in these populations? I'll go further and I say we don't even know. There can be all the selection in the world, but if there's a lot of gene flow, it'll homogenize the genetics anyway, and you won't see much of a QST difference. But anyway, we don't know which environmental axes are important. That's a bad thing, and it's because we want to know that. It's a good thing because we don't have to know that in order to apply this analysis. We can just say, oh, here's the trait that's interesting. We can go study the functional biology of this thing and figure out why it's doing this thing. We don't have to make preconceptions about what those environmental effects are. And then one thing I haven't mentioned, there's a real funky bias that builds into things like QST, and that is because it's such a large amount of work, you'd be insane to do this unless you already knew enough about your species to know which traits would be worth measuring. But if you know that much about those traits, you're going to be wanting to pick the ones that look like they're really different from one population to the next. And if you've gone through that filter of picking the ones that you think are diverse among populations, this is no longer the right null model. This is what all traits would look like. This is not like what all traits would look like if they were looking like they had enough difference to make you interested in them. I have no idea how to solve that problem, but it's definitely worth being aware of. You know how to solve that problem? No. Joe doesn't know. It means it's not possible to know. No.
It involved. It involves
not modeling biology, but modeling psychology of the investigator.
Right. Right. So here's some references that will help you with that. Now I've run over time. It's following the Steve model. So I have like 5 more slides on a different topic, which we can either totally ignore, or I can do now, or we can talk for a while and then come back to that. Or we can have no input on whatsoever, and I'll just stand here. Huh? Okay. Let's pause then. Anybody? I'm done with QST. Yeah.
You mentioned that, if one is going to endeavor to do QSTs, that it's important not to use a small number of populations, right? But say you're doing this in a greenhouse and you're limited on space. Should consideration be given, should you do, at the extreme case, 2 individuals per population as mini populations? Or is there going to be some kind of balance between number of individuals within a population and number of populations?
Yeah, there's a balance, and it's never been formally looked at. I've done some kind of just randomly curiosity-driven, just poke around and simulate it quickly and see. And something like the number of populations being similar to the number of individuals per populations seems to, something in the middle like that seems to work well.
Okay, and I got one more question. In terms of, so PSTs, I understand that they're not, they're kind of fraught with issues, but it certainly hasn't stopped people from doing them, right?
Yeah.
I guess in what cases would you ever be comfortable seeing them? So, there is this issue of environmental effects on the traits. If you had like a model of how environment affects traits and maybe include that in as a covariate when you're, estimating variance components, would you feel more comfortable, or are you pretty much just like never?
If you had a good model of that, it would depend on how good the model was, and then it would probably be a different thing that would have to be evaluated on its own rights. It's not impossible. So if you found a case where PST, let's say you had estimates of VA, but you didn't have estimates of the among population part, and you found a QST that was even then not different from FST, then you can be fairly sure that there's not good, strong evidence of spatially heterogeneous selection because it goes in that direction. And like I said, it's really difficult to interpret the opposite case where QST is smaller than FST, so it's not, you have to worry about it in that case. Yeah, I mean, another caveat, I'm not saying such information is not useful. I'm just saying putting it in the form of something like PST and comparing it to FST is not particularly useful. So knowing how much variance there is among populations and mean phenotypes is an interesting thing. And if you have that kind of phenotypic plasticity, it probably argues that there's some kind of heterogeneous selection that's causing that plasticity to exist. But that's not, that's a poor inference. That's not really what we're after. It could have not been spatially heterogeneous selection that caused it. Yeah. Is that?
From today's lectures, I've seen like there is kind of like looking at the adaptive landscape and the differences in populations. I'm kind of, I don't know. Can you like comment if we can use these kind of approaches to study? Let's not talk about speciation, but like population divergence. So there is a way of transitioning from tokagenetics to phylogenetics, if you want, like something like that.
So I hardly ever go deeper into the hierarchy than that. I mean, I don't think much about different species. So that's, I think there's a lot, like the kind of Brownian motion stuff that Joe's going to talk about probably shares a lot with this in some ways, right?
It does.
Yeah.
Yeah, they're direct connections.
Yeah.
Well,
You'll get there tomorrow.
Well,
Yeah.
we will be
Agonizing about that issue.
Right.
I should point out that there are members of Mike's family that do work on phylogenies.
There are members of my family who do, but that doesn't create familial knowledge that shares freely. But can we study divergence between populations? Yeah, absolutely. So I mean, there's a lot of techniques that people are working on now to look at genomic data, looking for outliers in FST, not just is this QST an outlier for that FST distribution, but is this particular locus an outlier? It's got its own challenges, but that's something we can do. You can do it old school. I mean, there's been a lot of I think the best evidence for local adaptation is is the kind of experiments that we could have done 100 years ago, like reciprocal transplants, where you move individuals in both directions in a carefully controlled way and look for what traits do well, do poorly. You can do selection analysis in different places, and if you can show that the betas are different in different places, that is evidence of divergent selection. Now, with that information, if you see that the differences in the populations are in those directions, you've removed half of the cases in the QST distribution that I've shown you, right? If it's not going in the right direction, it's not evidence of local adaptation, even if the selection is there. So when we start including information about the environment and how consistently do different populations differ from each other in a way that matches an environmental pattern, but importantly is not just the consequence of this sort of spatial autocorrelation that you naturally get in structured populations. What I mean by, then we can proceed. What I mean by spatial autocorrelation is that environments tend to vary in a way such that populations that are close to each other are experiencing a similar environment, and also the allele frequencies, because of gene flow being predominantly local, the allele frequencies are also spatially autocorrelated. And so when we start looking at such comparisons of environment versus gene frequency or environment versus phenotype, we have to be mindful of the fact that it's on a very complicated, autocorrelated, pseudo-replicated landscape and account for that in ways that everybody's still working on. But I'm very optimistic we can do it, but there's a lot of challenges. I'm going students first. Yeah. Because you're going to be mean. Okay.
So just going back to the spatially divergent selection with QST greater than FST.
Yeah.
So is it wrong to think of that as meaning that you can have divergence at quantitative traits even in the presence of very high gene flow?
Should I say that again?
So low FST, I'm guessing, so
Yeah.
QST greater than FST, which means sort of low FST,
Yeah.
which is high gene flow, I
Yeah.
guess you could. So basically it's saying you can have quantitative trait divergence even under high gene flow.
Yeah, true. So if you get low FST and high QST, that's the interpretation. Now, but remember what we mean by high gene flow. In an island model, the FST is determined not just by the rate of migration, but the rate of migration times the effective population size. And so it's the amount of gene flow relative to the amount of drift that there is that determines FST. So you can have, and so the complication is, and it's a good one, that whether selection can overcome the mixing caused by dispersal is largely a function of the strength of selection versus the effect of the migration rate. And that's not, and because that's not the only thing that determines divergence, happily we have cases where FST can be fairly low, but it's relatively easy for selection to actually cause differentiation. So large population size, low migration, moderate selection, you'll get low FST, but divergence for either loci or traits. And if that weren't true, we'd really be in trouble, because if it were strictly migration that determined FST, then exactly the times where we'd have any power to do it would be exactly the times where biology would prevent the local adaptation to start with. Yeah. Steve?
So if you go back to the PST, if you go back to the PST slide, so you're not going to always be wrong using PST. I'm going to give you an example. So in the garter snakes that I work on with vertebral numbers, I first started working on, I thought, there was literature showing that fish vertebral number is very much affected by temperature. Lots of experiments. And there were even some garter snake and other snake examples showing that this was true. And so I thought, once you hatch or, grow, once you're born or hatch, you keep the vertebral number through life. So anything that's going to happen plasticity-wise is going to happen during pregnancy. And the leading candidate was temperature. So I spent, several years doing constant temperatures and cycling temperature effects. And I planned on finding that there was going to be an effect and a maternal effects and so forth. And guess what I found? No effects over a huge. So so just because traits can be plastic doesn't mean they are.
Oh yeah.
And so basically, when I did a QST FST study, I didn't have any problem thinking and convincing reviewers that V among populations just probably isn't due to plasticity, because I cannot move the mean with a temperature treatment.
Right.
And that's what everybody thought was. And I also had a parent offspring estimate of additive variance for all of the traits I was doing.
Right.
So, I can't even remember what I called it, probably QST rather than PST.
Right.
But I look at those sources of inflation and think probably not true of my trait.
No, I think that's probably true, but you're bringing to it a lot more background about the species than sometimes people might have. And you're still making an assumption that nothing besides temperature was a trigger of the plasticity that might cause the populations to diverge. And so, so if you want to, I'm fine with that. I think that's I think that's good work. I'm just saying if you're trying to say here's a foolproof way of saying there is selection among populations effect, there's alternative interpretations, especially if you haven't measured this properly. Yeah, but you cleared up one of the obvious alternatives, so that's good job, Steve.
Your results may vary.
Your results may vary. That's right. Okay, so let me tell you about a little bit this other thing. It's been a while since I talked about this. So this is pretty much motivated by Steve's work, asking how stable is the Gmatrix over time. Is the Gmatrix a word you guys have used? Yeah. Good. Okay, I figured it would be. But, so there's been a lot of work from Steve and Adam, et cetera, and Reinhardt about how selection affects G. There's been a little bit about how drift affects G. But one of the things that hadn't been looked at much was how does putting the measures of Gmatrix onto a spatially explicit landscape, how does migration potentially affect it? So I'm going to use this term Gmax. It's just the first eigenvector of the G matrix. It's called Gmax because Dolph Schluter named this thing a few years ago in a paper in 1996. Cool paper you should look at. And what he said was, basically the hidden question, and I think it was hidden, maybe it was explicit, question of his paper was, everybody's doing all these measures of G. Should we care? Is there any useful evolutionary information there aside from G matrices being cool on their own? And this is just one of the figures from his paper, and he showed that, crap, I always screw up interpreting this figure whenever I get in front of people. Right, so he showed that especially when the time separating two context more, he went out to the literature from, 20 years ago, and found a bunch of different cases where people had measured a G matrix in at least one species or population and had measured the divergence between that species or population and some sister species or sister population, including, I think, garter snakes are in there, probably. Sticklebacks, Darwin's finches, et cetera, that kind of thing. So, right. So then he said, all right, for each of those G matrices, what's the main axis of G? What's this G max? And so if there's anything to this G matrix stuff, then it should be the case that the divergence between populations is skewed in the direction of G, right? So if selection alone determines the pattern of divergence between populations, then there may not be any signature of the G matrix in that divergence. But if there are axes along which there's not enough genetic variation to respond fully to selection, and other axes where there's more variation, then we should expect divergence to be biased in the direction where we have lots of variance against the direction we have less variance. Okay, so he said, let's compare the angle between Gmax and the axis of divergence between the two species. Okay. And so what he found, so zero's down here. Unfortunately, I'm sure Dolph made this in the early days of R before he learned to put the axes in the proper places. But the zero's about here. And that's where they'd be a perfect match between divergence and Gmax. This dotted line is what you'd expect by chance, okay, for that angle. And you can see that especially for younger divergences, less genetic distance between the species pairs or the population pairs, there is a strong bias in the direction of Gmax away from divergence. Okay, so I thought this was cool. He interprets this mainly in terms of G being predictive. He also points out very clearly that it could be that drift is more active along the axes of G. He doesn't think that's true. I don't think it is much either. He said that selection could in some way act to make G more like divergence. Okay? And that's a lot of the kind of things that Steve and Adam and Reinhardt have done, go a little bit in that direction. Or he mentions briefly it could be due to dispersal. And that final thing is what we set out to explore. This is with a former postdoc, Fred Guillaume. And so, long story short, we did a bunch of simulations of two trait models. And we basically started, so this population that doesn't have the red target in it is the ancestral population. So this is the G matrix of that ancestral population. And then we evolved them to 5 new optima. Okay? And those optima were aligned where the two extremes are 90 degrees different from each other, and then we put the other three evenly spread out between them. In other words, that's basically covering the full circle of where we could have gone, but because of symmetry, this is all the cases. Okay. Right. So this column over here is the case with no migration. Right. So you've taken this population, you've put individuals into this new adaptive landscape, and you watch evolution, but they no longer have any connection to each other. And our state. So this is no migration, and then these next columns are with increasing amounts of migration. The rows on this graph, sorry, this is a big bunch of stuff to be throwing at you. The top row, well, the different rows vary in other sources of correlation between traits. Okay, so the top, so row S is the correlation in selection between the traits, which is something like Steve's gamma 1, 2. Row M is the mutational correlation of the traits. How much does mutation alone make these things correlated? So the top one, there's no other source of correlation. It's effectively just a circular blob in the ancestral case, and without migration, each of the new populations is as well. Just there's basically, you can define a leading eigenvector for this, but it's extremely noisy. It really is not telling you anything about the truth of the biology. Right? It's basically an unstructured Gmatrix. In other words, saying that the correlations between traits are very, very low. When you have selectional correlation, that creates a correlation, a genetic correlation. When you have mutational correlation, that generates a correlation as well. Okay, but when you have migration, and let's just go over to the relatively strong migration case, 1% per generation of individuals come from the ancestral population to the derived population. I should say that better. Of the individuals in this derived population, each generation, 1% of them are coming from the ancestral population by migrants. You can see here is a case where there's no other source of genetic covariance. There's no selectional correlation. There's no correlation in the mutation function. But you get strong patterns of change in the shape of the Gmatrix that's a function of the migration. Okay? And when you put it like that, it seems like, yeah, sure, fine. It's actually a very intuitive thing. The migrants into this population here, for example, are coming from here. They're genetically like this. And so this population is a mix of individuals which are kind of like what they want to be for this population, plus individuals that are migrants or the recent descendants of migrants. And those individuals have genotypes which are going to be biased in that direction. Okay? So when you measure the Gmatrix of that, it's going to be stretched in that exact direction. And the same for any of these. There's nothing special in this case about any of the dimensions. It just gets biased. The resulting Gmatrix of those populations is just stretched toward the ancestral source of the migrants. Okay? But the effects can be fairly strong. Right? These are moderately correlated traits in these populations. These dots, by the way, are not genotypes. They're just a time series separated by I don't know how many generations each, following the mean go out. If you looked at the distribution of breeding values in that population, what it would discover is also that it's not, we didn't really talk about that in the paper, but you'll discover it's not Gaussian anymore. It's skewed toward the source of the migrants, as you'd expect, because there's, as I said a minute ago, there's recent migrants, there's the first back cross with migrants, there's second back cross with migrants, et cetera, and each of those is going to be more or less close to toward the ancestral migrant type. That persists even when we have other sources, strong sources of correlation between the traits, selection correlation or mutational correlation. Okay. So a big reason why G might change is migration, but more to the point, a big reason why you might get the divergence correlated with the shape of G is that ongoing dispersal between the descendant population and the ancestral population generates exactly that pattern. Okay. So if you have the possibility for gene flow, it's probably, I would say, that one of the strongest candidates for why you would get a co-alignment of G and divergence. Now, if you go back actually to, so Dolph talked about this in terms of time. Genetic distance actually isn't necessarily a measure just of time. Genetic distance is also reduced by ongoing dispersal, by gene flow, right? So these populations might have diverged just as long ago as these ones, but their genetic distance is lower because they have ongoing dispersal. So we can actually separate those two factors here. Okay. So that's, I think, the story. So if you have dispersal between diverging populations, you'll get greater genetic variance along that axis of divergence. And if you're not careful about it, you might misinterpret that as something else. So I should say, I don't know why, in the individual cases that Dolph talked about, why G and D match up. Any of the reasons seem plausible to me, but this one is not to be dismissed. Okay, that's what I was going to say. Questions? Yeah.
Any information or useful interpretation of the overlapping sections of the J matrices after migration? So do those overlapping regions mean anything? Like, I don't know, are they like more similar? I mean, we will expect that they are more similar because they share genotypes from the founding population, right?
Mm-hmm.
So, but there is like a formal piece of information that we can extract from those regions that are overlapping.
So when you say regions that are overlapping,
Like
G
in
matrix.
the actual ellipses of the matrices that are overlapping.
Oh, what can we infer from that?
Yeah.
Oh, that's a good question. I don't know. I mean, I think if we dug further, I mean, as we could do now, and actually get multilocus genotypes from those individuals, if it were the case that you were studying one of these populations, and you looked at individuals that were more like this in the descendant population, if that showed evidence of having a lower genetic difference at neutral marker sites from the ancestral, that would be a strong indication that it was probably dispersal mediated.
Okay, that's cool.
So yeah, I think it's entirely possible to at least try to pick this out.
And I have another question. So maybe I didn't understand the sorry. So I will just ask. So like if one trait of, or in this case, if one trait is selected for in the founding population, and for some reason the other trait is selected for in the divergent population, will you get like spurious trait correlations?
Well, in these cases, so both traits are selected for both places, but it's stabilizing selection around an optimum. I guess if you look, this one is divergent selection only on one trait relative to the ancestor, and this.
Oh yeah.
Yeah, and this one is only selection on the other trait relative to the ancestor, and this is correlated or selection on both traits relative to the ancestor.
Oh okay, yeah, I see. Cool.
That
Yeah,
what you
yeah.
can- Okay. Well, I just want to say, oh yeah, please.
I hate to ask this, but do you think you could go over the Schluter graph again? I got
God.
very, very lost. I'm sorry.
Yeah.
Yeah.
No, you just want to watch me squirm. Yeah. Okay. So here's Nay's genetic distance. Okay. So over here, the two populations are more similar for genetic markers, and here they're more different for genetic markers. And so down right about here is what you would get when the Gmax, the leading eigenvector of the Gmatrix, is exactly in the same orientation as the divergence between the populations.
I'm not sure if I understand what that means.
Okay. Can I erase this? I guess I can't. I got the eraser. It means I can't. Yeah, so if you had some just metaphor for drawing a Gmatrix, okay? Right, so there's just a bunch of Boolean values in here.
But you see, we should throw away some of
Oh, that's better. Yeah.
the sample after selection.
Right, so there's our Gmatrix, right? And so this direction is Gmax, right? So if the divergent, if this is before, here's the mean before, and here's the mean after, if that's exactly in the same line as Gmax, that's what it means here of a zero divergence. And I guess these are in radians. I can't actually tell you what 1.2 is intuitively, but anyway, it's a little, yeah. One of the things I'm confused about this is the different G matrices had different numbers of characters, and so the expectation was different in different so when he actually did it, it was used the correct random deviation. And I don't know what he did here. Maybe he reduced it to trait by trait, 2 trait cases. I don't know. Read the paper. Yeah. Yeah. Do you have a...
So I'm just wondering, the Gmax here, it's calculated on what? So this is the G matrix of the, because we're talking about two population, about a pair of populations. So this is the Gmax on the
on one of the populations.
On one of them.
Yeah. So
right.
All the data that he had in this paper, somebody had measured G for traits they cared about that they thought were important ecologically on one of the species, and then on the other species they only had morphological means.
Okay,
right.
So this is sort of a random pick of one of a pair, in a way, or
yeah, randomish probably. I mean, it's probably the one that was easier to grow or easier to get or whatever. But yeah, hopefully random with respect to the other properties.
Okay.
So in this, you're measuring the orientation of the first eigenvector in the two. So if there's a matrix with larger number of traits, it'll like with 3 dimensions, could choose any angle that it wanted to in 3 dimensions.
Yeah.
And in 2 dimensions, any angle in the circle or half circle.
Right.
And though the averages of those are different.
Yeah.
So that's why there was this problem of...
Yeah, so the main bulk of the paper, not necessarily what I chose to show you guys to make the point. The main bulk of the paper, he said, well, this one has 5 traits. So for 5 traits, he figured out if it was totally random in direction, you know, full hypersphere direction of where it went, what would that theta be? Yeah, and then compared it to that with Bootstrap.
Okay.
Yeah. Oh, there was another hand a minute ago. Okay. Well, I was going to say, I'm here all day and then maybe in the morning too, and I'd love to talk to anybody who can stand talking to me. So if you care about any of this stuff or just want to talk about anything, I'd love to hear what you're doing, anything. So just, I'm around. Come find me. Yeah. Anyway. So thanks.
Thank you, Mike.